First, I'm going to tell you why we care about trace metals. Then I'm going to tell you about remineralization and how we try to bring remineralization from the ocean into the lab. Then I'm going to go through my methods, both the experimental setup and some of the analyses that I did. Then talk about my results, and then finally sum it up and talk about some broader applications of this research. So why are trace metals important? Well, this figure here shows the concentrations of some of the elements in seawater. On the y-axis, it shows the concentration of these elements listed above, or in approximately the relative amounts listed above, with iron being the most abundant, followed by zinc, manganese, nickel, cobalt, copper, and cadmium. So why are trace metals important? Well, phytoplankton use the trace metals that I listed in a variety of different roles. Iron is participate in electron transport reactions or electron transport chains, and participates in photosynthesis and the nitrogen cycle. Zinc plays a role in alkaline phosphatase and carbonic anhydrase. Nickel plays a role in urea metabolism in the enzyme urease. Manganese is utilized both by phytoplankton as a part of superoxide dismutases to protect against oxygen radicals that are generated during photosynthesis, as well as being utilized by manganese oxidizing bacteria at depth that can form manganese oxides. Cobalt is a cofactor in vitamin B12, which we as humans need and phytoplankton also need. And it can also substitute for zinc in carbonic anhydrase. Cadmium can also substitute for cobalt or zinc in carbonic anhydrase. Copper is often called copper has a hybrid type profile. It is sometimes called a Goldilocks type element in that it can either be toxic or a nutrient depending on the concentrations, and it can also be co-limiting with iron in some situations and can substitute for iron. And finally, lead is not a nutrient, but it is commonly scavenged, so it's still useful to look at. So we know that trace metals are vital for life in phytoplankton. But what happens after the phytoplankton die? And the answer is remineralization, also known as regeneration. So how does remineralization affect the concentrations of trace metals and macronutrients in the water column? Well, first of all, in the surface ocean, where there's light, phytoplankton take up both macronutrients like nitrogen, phosphorus, and silicon, as well as the trace metals. And then when the phytoplankton die and sink and decay, they release the macronutrients and trace metals in a process known as remineralization. And this process helps influence the concentrations of both the trace metals and the macronutrients at depth in the water column. So here are a few profiles of both trace metals and the macronutrient phosphate. You can see for phosphate, in the surface ocean, it's very low and depleted, then it increases and then kind of stabilizes at depth. And this is a very classic nutrient type distribution. However, for trace metals, it's a little bit more variable. For example, for zinc, here are a few of the trace metals. For zinc, it tends to have a profile that looks very similar to macronutrients, such as phosphate and especially silicate. However, for some trace metals, such as cobalt, the profile looks quite a bit different. It's higher at the surface, then decreases with depth. And for others, such as iron, it can be influenced by both nutrient processes and other processes, such as scavenging. And so trace metals are variable. They have a lot of different factors influencing their concentration in the surface ocean. So the goal of this thesis is to look at the process of remineralization in the lab and see, okay, how does that contribute to the concentration of trace metals that we see over time, and how can that then be applied to what we see in the water column? So how do I bring remineralization from the ocean into the lab? Well, the first step was to collect seawater, then to induce a phytoplankton bloom, then to kill it. Put the phytoplankton in the dark and let it die over the course of 6 months, and as it decays, watch what happens. So over the course of 6 months, I took regular samples for both parameters relating to biomass like chlorophyll A, imaging by scanning electron microscopy, particulate organic carbon and nitrogen, dissolved macronutrients, which include N plus N, which means nitrate plus nitrite, in addition to nitrite alone, ammonium, silicate, and phosphate, and the trace metals. And I looked at manganese, iron, cobalt, nickel, copper, zinc, cadmium, and lead. The key questions I wanted to answer are, 1, how does the timing of dissolved trace metal remineralization compare to that of macronutrients and other trace metals during phytoplankton decay? And 2, how do the trends in these remineralizations compare to ocean depth profiles? So how did I do this? Well, I set up a total of 5 remineralization experiments, and I'll be using this notation throughout the rest of the talk. So M2, M1, M2, and M3 stand for mixed assemblages. So for these, I took unfiltered seawater from the Gulf of Mexico, which had a mix of different phytoplankton in it, and grew it and induced a bloom. P1 stands for Pseudonychia dolorosa, and this was a monoculture that was grown at FWRI and then added to a carboy of filtered seawater. K1 stands for Carenia brevis, which is a dinoflagellate, which is also a monoculture grown at FWRI and then added to a carboy of filtered seawater. Whoops. So for the mixed assemblages, I collected the seawater fairly close to shore, so it had enough phytoplankton in it to get a high biomass bloom. For M1, I collected it by hand by submersion filling a carboy. For M2 and M3, I used a towfish off the side of a ship and collected it in a clean air bubble. In both these cases, I did my best to minimize possible metal contamination from the ship and did everything in as much of a trace metal clean environment as possible. For P1 and K1, again, I collected filtered seawater and then only later added the cultures to it. So I did this much further offshore, kind of right off the Florida escarpment. And this was very low nutrient, clean oligotrophic water that was filtered shipboard and taken back to the lab. For the mixed assemblages, it was necessary to perform a grow out to achieve high cell biomass. So I added iron and macronutrients. For M2 and M3, I added iron 57 as the source of iron, and this was simply a tracer to track what iron was taken up and released by the cells. I then induced a bloom in a light and temperature controlled environment, which took about 2 weeks, and then afterwards covered the carboys in dark bags and let them remineralize over the course of 6 months and took samples along that time period. For P1 and K1, I did not perform a grow out because the cells were already cultured at FWRI separately. For the Pseudonychia, we filtered this cell culture by vacuum filtration to make it as concentrated as possible. For the Carenia, because the cells were more fragile, this wasn't possible, so we just added the concentrated cell culture directly to the carboy. Again, we added these to carboys of filtered seawater and then put them in the dark and let them remineralize over the course of 6 months. For all of the carboys, all 5 of them, once they were in the dark, I sampled at regular intervals with higher resolution near the beginning of the sampling period. So I sampled at roughly 0 days, 1 day, 3 days, 7 days, and then monthly thereafter. As I mentioned before, I sampled for parameters relating to biomass, dissolved macronutrients, and dissolved trace metals. For the purpose of this talk, I will explain in a little more detail the analyses that went into looking at the dissolved trace metals. But I will give you the results of everything. For the trace metals, it was necessary to use an instrument called CFAST, which has something called a chelating resin. The reason for this is that in seawater, you have a ton of salts like sodium chloride, and your trace metals are only at very, very low concentrations. And so in order to measure the trace metal concentrations on an instrument like our inductively coupled plasma mass spectrometer or ICP-MS, it was necessary to separate those trace metals from the saltwater matrix and to make them more concentrated. And to do this, to prepare the samples, I first filtered the seawater through a trace metal clean filter rig. I acidified and let it sit for a minimum of about 2 weeks, but often longer, and then UV oxidized. And the purpose of this was to free some of the trace metals, particularly cobalt and copper, from the organically bound ligands, which can cause the yield to decrease if they're bound to ligands and they pass through the chelating resin. After that, I performed pre-concentration using an instrument called a CFAST, which is basically just an automated way of running your trace metal samples through this chelating resin. First, the samples were buffered at a pH of 6.5. They were then run through the column, and there are chelating groups on this resin, and so the trace metals stuck to those groups. And then afterwards, the CFAST rinses with 5% nitric acid and indium, which is an internal standard, and elutes out that concentrated solution of trace metals. And I generally pre-concentrated by a factor of about 20, so it got 20 times more concentrated than it was and was separated from that saltwater matrix. After I had those solutions, I then ran on an ICP-MS and normalized to the indium internal standard and used a calibration curve that had also been run on the CFAST to get the concentration of those trace metals. And I looked at all of the trace metals that I listed above. So what do we find from this? First, I'm going to talk about chlorophyll A, probably the simplest of the results, which is a proxy for biomass. This basically tells me, okay, did the phytoplankton grow? Did the phytoplankton die? I'm going to first explain a few things about these figures because they will carry through for the rest of my presentation. So pay attention. For M1 and M3, recall that these are the mixed assemblages from the Gulf of Mexico, unfiltered seawater. I will be showing these on the figure on your left. M1 is on this axis, M2 and M3 are on this axis. For P1 and K1, I will be showing these on the figure on the right. Again, P1 is on the left axis, K1 is on the right axis. It stands for pseudonychia, the diatom, and Carinia, the dinoflagellate. Also note that the y-axes are of different magnitudes. This is just because for my cultures, I had they had different amounts of cells, different amounts of trace metals and macronutrients, so it was necessary to make the y-axis different to make it easier to visualize. Also note that the x-axis is split at 10 days, and this is just to make the resolution at the beginning part easier to see. Another thing you'll notice is that for the mixed assemblages, recall that it was necessary to perform a grow-out. The grow-out is what is shown in yellow here, and it's represented by negative numbers. So negative 14 is the first day when I collected when I emitted the seawater before it had grown. Day 0 is the day that it had finished growing and I put it in the dark. And the gray is the dark part. For P1 and K1, I did not perform this grow out, so there is nothing in this space. It just starts at day 0. So now that we got that out of the way, what did we find? Well, from approximately days 20 onward, for all the assemblages, chlorophyll went to almost 0. So that's what we expected. That's what we were hoping to achieve. It means that there are no longer living photosynthetic cells. I also looked at particulate organic carbon and nitrogen. For particulate organic carbon, what we saw is that the relative amounts of particulate organic carbon in all of the assemblages, in all of the incubations, roughly followed what we saw with chlorophyll. In addition, even though it was a little bit messy in some, like P1, the general trends that we saw was a decline over time as the cells died. So that's what we expected. We also looked at PON, which because nitrogen is in the cells in lower concentrations than carbon, it was below the limit of detection in a lot of the time points, but for the beginning part, at least in M2 and M3, we saw a trend that was generally similar to POC. I also looked at the diatoms in M2 and M3 by scanning electron microscopy. We did not collect samples for this for M1. However, based on the growth that we saw and the uptake of silicate, for example, we believe that it was also diatom dominated. For both M2 and M3, for the first 7 days, we saw that both assemblages were predominantly diatom dominated. They had primarily pinnates and also some spores, which you can see here. And then by 124 days, we saw a lot more diatom fragments, which is what we expect. The diatoms are decaying. We also saw placcoliths from the coccolithophore, Gephyra oceanica. And in tongue-tied, in M3, we saw some more of the heavily silicified diatoms that were still intact, like Odontella, for example. So we did see some differences in morphology in M2 and M3. However, we could still tell that they were from the same seawater, and both were predominantly diatom dominated, and the trends were similar. So how did the macronutrients agree with the story that we're seeing of SEM and POC and chlorophyll of diatoms decaying over time? Well, first we'll look at phosphate. Phosphate, as you can recall from the water column depth profiles that we saw in an earlier slide, in the water column is depleted and then increases with depth over time. And so what we're seeing is not a depth profile, but a sort of time profile that's horizontal instead of vertical. And so what we see ends up looking pretty similar to what you would see in a depth profile, in that our phosphate is at the lowest concentration at time 0 in all of the carboys, and then it gradually increases over time. So that's what we expected, and that's encouraging. It's showing, okay, our macronutrients are behaving basically how we expected them to behave. We also looked at silicate, which showed no decoupling from phosphate. It was very closely coupled, and in everything except K1, Carinia, which of course is a dinoflagellate. It does not have silicate frustules, so we would not expect to see much of a change. So that's what we expected too. So that was encouraging. We also looked at nitrogen species, N plus N, which is nitrate plus nitrite, nitrite alone, and ammonium. A bit of background first. In the 1930s, von Brand et al. Did a series of classical remineralization studies where he put phytoplankton in the dark and watched them decay as well. However, von Braun did not look at the trace metals. He just looked at nitrogen and looked at the nitrogen cycle. And so what he saw was this nice peak for ammonia, or this nice increase in ammonium. And then when ammonium went down, he saw an increase in nitrite and then in nitrate. So it's this very predictable nitrogen cycle. So the next question is this what we saw? So for ammonium, we saw an increase in ammonium in all the experiments, which is what you would expect to see is an initial increase in ammonium. For M2 and M3, ammonium declined to essentially 0 by the end of the experiment. You can't see the data point for M3 because it's hidden beneath M2, but they're both at 0 here. For M1, P1, and K1, we also saw an increase in ammonium. However, it did not go down over time. So the next question is, well, did nitrite and nitrate agree with this? Based on this data and based on what von Braun et al. Did, we would expect to see an increase in nitrate and or nitrite around here for M2 and M3, but not really for the rest of the assemblages. And so for nitrite, what we saw is we had a very distinct increase for M3, a slight increase for M2, P1, and K1, and not really much for M1. We also looked at nitrate plus nitrite. So this is essentially, you can get nitrate by just subtracting the nitrite from the earlier slide. We only really saw an increase, again, in M3 in addition to M2. So it really only looks like nitrification is happening for M2 and M3. So why is this? Well, one reason that could be attributed to no nitrification happening and the persistence of ammonium in some cases could be that your water went suboxic. However, this was not, we do not believe that this was the case because one, we had residual nitrate, we had residual N+N in M1 as well as nitrite species in K1 and P1. In addition, I checked the oxygen levels regularly and they were all above 97%. And I bubbled oxygen into everything except M1. So it is not likely that we went suboxic or anoxic. More likely, the explanation is that we just didn't have nitrifying bacteria in all of the experiments, which makes sense because M2 and M3 were collected from very similar, or from the same water source, but M1 was collected from a different spot, and K1 and P1 were just the cultures added to filtered seawater. So most likely, they just didn't have the right bacteria in them to cause the nitrogen cycle to go to completion. But for everything else, all the other nutrients, phosphate and silicate, they proceeded basically how you would expect to see them in the water column. So that was encouraging. On to the trace metals. Everything we've talked about here so far leading up to the trace metals. The reason we looked at the macronutrients is to basically see, okay, how do these trace metals compare? So this is the exciting part. First, let's talk about cadmium. Up at the top, you'll see this looks familiar. It's phosphate again. And at the bottom is cadmium. And they look very similar. And in the water column, if you remember what if you're familiar with water column profiles for phosphate, it looks very similar to or if you're familiar with the water column profiles for cadmium, they tend to have a very nutrient type distribution. So this was very much like what we would expect based on water column profiles. So cadmium resembled both phosphate and silicate. We also looked at zinc. Up here is now cadmium, and this is zinc. So zinc again increased over time. And in the water column, zinc also tends to have a very nutrient type water column profile, although zinc tends to be more closely coupled with silicate than with phosphate. However, zinc was not quite as closely coupled with either phosphate or silicate in our experiments as cadmium was, but that could be partly because zinc is just more prone to contamination than cadmium is. But overall, zinc had a general nutrient type trend as well. Manganese was very interesting. In the water column, manganese is known to be influenced by manganese oxidizing bacteria at depth, which are photoinhibited by light. And so we would expect in these experiments that if we have manganese oxidizing bacteria present, we would expect them to oxidize our manganese cause it to precipitate out of the water and therefore cause our concentration of dissolved manganese to decrease. And this is what we saw in all the experiments. In some of them, like in M3, M1, and P1, you did see an initial remineralization signal where there's dissolved manganese increasing, but then in all of them, manganese eventually declined to lower levels. And we believe this is evidence of manganese oxidation. Iron, as I mentioned previously, has a hybrid type profile. It can be influenced by both scavenging and nutrient uptake and remineralization. And so we would expect that if manganese oxides are forming, iron would most likely be scavenged to them. And what we saw is that iron had a delayed remineralization signal in all the experiments except P1, which did not show a remineralization signal at all. It just kept on decreasing. However, eventually, iron did start to increase. And when we looked at M2 and M3 iron 57, the magnitude is different, but it follows the same general trend where you see a delayed increase. So why is this? Is it simply being slower to remineralize, or is this a combination of scavenging and remineralization? Well, one thing that's cool is that we noticed that our data looked very similar to another study that just recently came out of the Fitzsimmons lab by Chitse et al. And what they saw, they did a remineralization study of biogenic sediments, which are shown, or biogenic silicate sediments, so predominantly diatoms, and they're shown in these green diamonds. And what they saw was also a delayed increase in iron remineralization, which is approximately at the same time that we saw it. They also looked at manganese, and what they saw is that manganese decreased, and then stabilized right at around the point that iron started to increase, which is also what we saw. And so the reason, and they saw evidence of manganese oxidation by manganese oxidizing bacteria, and so the reason given for this, the reason proposed in this paper was that iron remineralization is happening from the beginning, but because of manganese oxidation, iron is being scavenged to those manganese oxides, and then only after manganese oxidation stabilizes can you start to see that remineralization signal from iron. So that was really cool that our experiments looked so similar to theirs, and it kind of validates that what we're seeing is real. We also looked at lead. Lead is primarily influenced by scavenging in the water column. However, unlike iron, we wouldn't expect to see much of a remineralization signal from lead, which we didn't. Lead basically decreased, and in some we see a slight remineralization signal, possibly as the cells are decaying, that it's being scavenged to. However, it did not show that same delayed remineralization signal that iron did, which makes sense because iron is an important nutrient, lead is not. And finally, copper, cobalt, and nickel were all largely conservative in the experiments. Copper, however, there are a couple trends that I want to point out. Copper, for example, in M2 and M3 looked kind of similar to iron. It had that same delayed remineralization signal where it stays fairly low up until about 60 days or so, and then it starts to increase in M3, and then a little later it starts to increase in M2. So that remineralization signal that we saw in iron. And copper is known to have a hybrid type distribution in the water column. It's influenced by both nutrient influences and scavenging influences, so it makes sense that it would be influenced by similar things as iron. Cobalt is known to be influenced by scavenging to manganese oxides. However, we didn't really see much evidence of this in this experiment. What we did see is that in M1, both cobalt and nickel seem to have more of a nutrient type distribution where they had a drawdown at the beginning and then increased. And then it's the same for nickel in M1. Drawdown and gradually increase a little bit. But in all of these, it was the change was relatively small relative to the overall reservoir. So what do we learn from this? This is a lot of data. There are a lot of trace metals that we talked about here. How can we sum this all together and not be overwhelmed? Well, let's just sum up some of the main points first. Silicate and phosphate remained closely coupled. Cadmium of the trace metals was the most closely coupled to the macronutrients. Manganese showed evidence of initial remineralization, followed by loss to manganese oxidation. Iron and lead both showed evidence of scavenging to manganese oxides, followed by a delayed remineralization signal for iron. Copper resembled iron the most closely in M2 and M3. Cobalt and nickel both displayed more nutrient-like uptake and remineralization patterns in M1. And for the rest of the experiments, copper, cobalt, and nickel were otherwise largely conservative. So the next question is, okay, we have this data from the lab. How can we apply it to the water column? So if you look at our cadmium profile, for example, this is the increase over time. And if it looks a little different from what I showed you before, it's because I'm not using the split axis here. This is just to make it easier to compare to the water column. So cadmium is lowest at the beginning, then increases. And this looks a lot like the depth profile. If we flip this clockwise, 90 degrees, looks a lot like cadmium remineralization at depth. So one thing that can be useful is to take actual depth profiles and compare it. There isn't really this trace metal data for the Gulf of Mexico. However, if you go to egeotraces.org, there is geotraces data available online. And so I decided to take some of that data and graph it. And so I chose a station kind of nearby Florida, similar latitude, and decided to graph these data to compare them to my data. So the geotraces data is in blue. My data turned on its side for M1, M2, P1, or M1, M2, and M3, P1 and K1 is right here. And it's just kind of a side-by-side comparison. And you can see for cadmium, it looks pretty similar. It's not exactly the same. You may have some deep water influences here, but in general, it's highest at it's lowest at the surface, just like it's lowest at T0, then increases. Zinc, again, looks pretty similar. Lowest at the surface, increases with depth. Lowest at time 0, increases with time. So that's encouraging. Manganese is totally different. You can notice for the geotraces data, it's highest at the surface, then becomes depleted. And it's the same in our experiments because of manganese oxidation. And then iron is complicated. We don't have exact, like our data does not look exactly like the geotraces data, but if you were to take an iron depth profile somewhere else in the ocean, it may look different because iron has that hybrid type profile. It can be influenced by both nutrient influences and scavenging, and we see evidence of both. And so iron is complicated, but we believe we got useful information on iron and manganese oxidation from our experiments. So in conclusion, we're hoping that this data that we collected from the lab can be eventually applied to the bigger picture and utilized to help us better understand the behavior of trace metals in the ocean. I'd like to thank everyone that has helped in the lab. First of all, our funding sources, Tony for his beautiful SEM images, Shannon for her help with CFAST, Salvo and Kelly for their help with ICP-MS, Jen for letting me use her fluorometer, Joe for helping with the environmental chamber, Ethan for the particulate organic data, Katie for helping with the burger leeches, which I didn't show here, but they are in part of my written thesis. Celia for her help with all the phytoplankton SEM identification, everyone for their help with seawater collection, and of course all my lab mates, Shannon, Gabe, Travis, Chelsea, Casey, Will, and Salvo.
We will open it up to questions from the audience. I'll let you call it.
Okay. You're going to make it easy for me. Yes?
What would be the expected remineralization at deeper depth?
That's a good question. So obviously, this experiment did not account for changes that would occur with depth. One thing that you could expect to be different is that you may have different bacterial assemblages. You would also expect to have different or increased pressure and decreased temperature and decreased or changes in saturation state. So I wouldn't expect the conditions to be exactly the same at depth. You would also be mixing from deeper water at depth as well. So you would, that is one potential weakness of this, is that it doesn't perfectly replicate the water column, but it does allow us to isolate this process of remineralization that's happening without the other influences.